Dr. Agarwal.
So nice to see you, sir.
You are from your home.
Yes, Dr. Kalra, we can start. We are live on YouTube. We can start.
Thank you, and I welcome Dr. N.K. Gongli, the professor, and all other distinguished participants who have joined. Still, some are in the joining process. It's my duty first, although Dr. Gongli is a today invited guest, he needs no introduction. He's a well-known, but still, as a customary, I will just briefly introduce him. Dr. Professor Nirmal Kumar Gangli is a former director, ICMR, Dr. Journal, and he is presently visiting professor of Eminence Policy Center for Biomedical Research, Transitional Health Science and Technology Institute, Faridabad. He is also honorary senior research professor, clinical research, Institute of Liver and Biliary Sciences, New Delhi, and the professor attached to Gangaram Hospital, as well as many other prestigious institutes. He has published more than 775 research papers and has supervised 130 PhD theses as supervisor or co-supervisors. He's a member of advisory group. So many national and international groups he is a member, which if I go on for 2 hours, probably this will not end. And also he is an advisor to regional director CRO and is a member of regional task force on disease targeted for elimination. Dr. Gangli was selected for award of Helmholtz International Fellow by Helmholtz Center for Infection Research, Germany, for a significant contribution to the field of infectious disease. And also he is a Padma Bhushan awardee, year 2008, for the field in the field of medicine. Now, I welcome Dr. Gangli to this forum. I also welcome Dr. Arun Kumar Agarwal. He is a former Dean of Manasa Medical College and our Chairman Ethics Committee, Healthcare Foundation of India. And also welcome Dr. Snila Agarwal, Dr. Alex, Dr. Sanchita, and Dr. Jayakrishnan and Mrs. Upasana Arora. With these words, now I invite Dr. K.K. Agarwal to give introductory remarks, and then we invite Dr. Gangli for his.
Dr. Kalra, this subject is of utmost importance.
Yes.
We have had interaction with Professor Gangli on 2 previous occasions, and but every week, every month, the vaccine update needs to be updated.
Yes.
The reason being, today most of the countries also wants to improve their economical status. And we all know that whichever company brings the first vaccine will first improve their economy and economical status, because it will be a trillions of dollars industry on day one. So therefore, we have to be very careful that in the run of fight of an economical improvement, health should not be compromised.
Yeah.
Recently, there was a statement given by internal document that ICMR is launching its product by day 15th of August. Probably it looks like is that they probably want to compete with Moderna and with the Oxford, and they want to finish phase 2 trial, so that in a phase 3 trial, they are in parallel to the Moderna and to the Oxford group, so that they can compete with the race. This is what it looks like. And I don't think so there was an intention of launching a commercial project by 15th of August. Practically impossible. And the second issue is that can ICMR be an interested party in a commercial game where ICMR is both the regulator as well as the commercial interest? And if yes, in future, all such expedition should occur in all clinical trials, in all vaccine development, and all research is in India, I am a game for that. That means if ICMR and DCGI starts giving approval to every clinical trial with a deadline, and all trials are completely the permissions given with a deadline, India will reach the height much earlier than we are thinking today. I don't want to go into the basics of the vaccine because that's a field of Dr. Ganguly. So let's listen to him. And after that, we will, even if we have to extend our time a little bit, we will, because that's an important subject for today. Dear colleague, I present to you Dr. Ganguly. This session is live at multiple social platforms, and I expect that this program will be listened live by not less than 10,000 people. So this is just for information that we are at least 10,000 people might be getting connected to this program. Thank you, Dr. Ganguly.
Thank you, Dr. Agarwal. I don't want to get into that ICMR and ICMR letter. It was mostly, as you have said, aspirational. They wanted that by 15th August, the first shot to human for the phase 1 trial should happen. But there was no deception in the CTRI. It was written, the time frame was written, and for the phase 1, it was written 3 months. And then the phase 2 was written. This also takes about that time. And there was no mention of phase 3. So I don't think there was any deception. It was aspirational, and it went on to all kinds of controversies. The controversy is settled now. The Health Ministry has made a statement that it should be out by 2001. And today, Dr. Harshvardhan was in ABP News, and I was looking at that. He also said that we cannot commit that when we will have a vaccine. I totally agree with him. It is very difficult to predict that when you will have a vaccine, because there are more than 100 something vaccine are being made by different people, and 90% of them will fail. They will not see the light of the day. Few of the vaccines might succeed. It's not a very easy thing to do. So if you really ask me today, we don't have a vaccine, but every one of us wants a vaccine, because to build a herd immunity, it takes very long time. In United States, in some areas now, the herd immunity has reached 25%. But until unless herd immunity reaches 60 to 70%, you cannot diminish the spread of the infection in the community. So, but if we get a vaccine, this is possible. There are different scenarios in the vaccine thing. The vaccine may protect 40%. It will not be an ideal vaccine, but even then it will protect some people. Maybe it will be used. It may protect 50%, but produce for 50% protection. Even then it might be okay. But if it protects 70%, it will stop the transmission. There will be herd immunity because of the vaccine, and it will stop the transmission. So nobody knows at the moment, because only trials which have happened are phase 1 and phase 2, and the phase 3 are yet to be done. So 19 of the vaccines around the world have completed phase 1, or are in phase 2, or have completed phase 2, and they are going into phase 3. So they had started it much early. And so some of the international vaccines are in the phase 3, and they will come out much earlier than the vaccines which we are attempting over here, because our vaccines are just entering safety studies in phase 1. However, the animal toxicology studies for the vaccine is not an easy thing to do here, because although there is a WHO guideline for animal toxicology in vaccine, which is much easier than those of the drugs, but for the COVID vaccine, there are issues. And that is why US FDA has brought out a separate guideline for this vaccine. So for this vaccine to qualify, you have to really do two studies, which most of the vaccine companies have done, and two of the Indian companies have also done. This has to be done in a transfected mice with adeno ACE2 vector, which makes this mice ACE2 expressing mice. And then you then you immunize them and challenge them with the COVID-2. And if they're protected and if they form neutralizing antibodies, they are okay. This can be done even in Barbsey mice. The second one is the monkeys. Monkeys have an infection. They develop an infection when given COVID, and you have to do a protection in monkeys. And monkeys should also have neutralizing antibodies, but monkeys should also have 2 or 3 different characters, because and as the dengue vaccine or the respiratory syncytial virus vaccine and some other vaccines have produced immune reaction against itself, against the person who is receiving it, the same happens in the COVID. Theoretically, it happens, and it might happen. So that is why you have to assess that whether there are self-reacting immune responses which have been generated or not, and this is essentially seen in the monkeys. So both of these are very tedious processes and they take time. Then another thing which has to be done, that they need to interact with the plasma, pooled plasma antibodies, so that at that time the vaccine response is the vaccine construct is relevant. So to make the vaccine safe, there are different platforms which have been used, with which the companies had previous experience. And those vaccines which have moved faster, there the companies have already used these platforms for other infections, such as influenza vaccine, such as Ebola vaccine, such as RSV vaccine. So they have used for other RNA viruses such vaccines, and they already have an experience that how this platform does and how the adjuvants, etc., do. So some of the major vaccine companies of the world, they have these. And to make the vaccine safe, they use very limited amount of viral proteins. Like many use a preformed spike protein. Many used even a small amount of the RBD, small motif of the RBD, that is the receptor binding domain. So, and make it as safe as possible so that the vaccine doesn't have other viral components which might create a problem. Whole cell vaccines may have a problem, viral vaccine, whether they are attenuated or whether they are killed, because they have all other viral antigens which might produce immune responses. So the preclinical toxicology takes time, and it need to be done absolutely world class if we want to make this vaccine move. So at this given moment, if I take, because in the world there are several vaccine candidates which have moved faster, and many of them are now go for the phase 3. They are moving to Brazil. Because Brazil has got the very strong infection, it will be easier and smaller study for the phase 3 if they move to Brazil. And then there may not be enough vaccine made which will cover everyone. So the immune responses in the humans, they differ from different class to class. So that is why when these vaccine trials are being done, they are divided into 3 different segments. One segment is 18 to 50 years or 55 years. The other one is 55 to 70 years, and the third one is 70 years and above. So there will be two different scenarios who will receive the vaccine. One, who are old people like us and who have pre-existing conditions, so we are vulnerable and we will need a vaccine. So if the vaccine is available, we will be one of the candidates who will receive the vaccine. So it will be good to put test the vaccine in this particular age group also. Normally they start from 18 normally they start from 18 to 55 years of age. So 18 to 55 years of age will not qualify us to receive that particular vaccine, because if our age group is not taken, it will not be possible for our age group to give that particular vaccine. So simultaneously, all the groups are being, three age groups are being done, and people are taking different concentration. That is determined, finalized in the phase 2, but different concentrations are taken and multiple injections are needed. At least 2 injections are needed, one to prime and one to boost. So this is the vaccine development scenario. Very difficult, very unpredictable. So what at the moment is taken as that vaccine will work or not is that whether there are neutralizing antibodies formed or not. Neutralizing antibodies are checked, they are very difficult assays. They are checked in two particular format. One is the live virus and other is the host cells. So it will be exceedingly difficult to the validated neutralizing antibodies are not there. So, so whosoever got neutralizing antibodies, they claimed that they have succeeded in getting neutralizing antibodies during the phase 1, as well as the safety of the vaccine in the phase 1. So Moderna got neutralizing antibody in its first group. In 8 of the subjects, it got neutralizing antibodies, and it also passed the safety. Safety means that there will be swelling, there will be fever, there will be redness at the site, there could be some nausea. These are taken as a these are not really serious adverse reaction. Serious adverse reaction will be that if the vaccine produces a cytokine storm, if it produces autoimmune reaction, and if it attacks. At the moment, it has not happened with the Moderna vaccine, because Moderna vaccine is a synthetic vaccine, and this is a synthetic. It is not, there is no virus in there. So this synthetic vaccine has been put in a carrier, and with an adjuvant it is given. So Moderna has moved to the second and third group, but Moderna has also finished the second phase in 650. But they haven't really published the paper. Paper have been not published in peer-reviewed journal. Only paper for the phase 1 which has been published is by the Sinovac, that is a Chinese company. They are doing their studies in China, Pakistan, and Brazil. So let us take this messenger RNA vaccine. The Pfizer is also taking 4 different constructs of messenger RNA vaccine with BioNTech, and 2 of them are moving, and they are moving again. They have completed phase 2 and they are moving forward. Pfizer has got a lot of experience. It purchased Wyeth and other vaccine companies and has a lot of experience with their platform. And BioNTech is producing that other major capability. In India, the Moderna has not Moderna, the Genova is developing a messenger RNA vaccine. They are also very much advanced. They are almost finishing preclinical studies. Their results in the 2 monkeys are excellent. Their result in the mice challenge is excellent. They have completed their study on the plasma, convalescent plasma. They find that it is very nice. They did in their vaccine, and in modern, like Moderna and Pfizer vaccine, they didn't find that there is any autoimmune reaction or cytokine storm or deleterious immune reaction. However, even if these vaccines go to human, there is no certainty. I want to say that till the phase 3 is done, there is no certainty that whether the vaccine will succeed or whether the SAEs will have to be, and different regions may according to their endemicity may react differently. It happened in the rotavirus vaccine. It worked in United States, it worked in Europe, it didn't work in Malawi, it didn't work initially in India. It did work in many countries where the infection was already endemic. So we can't say that any of these, but these are the best at the moment. This thing at that, but there is a but. There is no messenger RNA vaccine in the world which is there in the market. So the entire actually experience will have to be harnessed. And that's why I say, and I am glad Dr. Harshvardhan made this statement today, that we do not know when we will get a vaccine. So this is one candidate. So in this candidate, Moderna and Pfizer have gone into humans. The Genova hasn't been able to go into human, but they are almost completing the animal and pre-toxicology uh, studies, and both proof of concept studies. And their monkey challenge studies, they claim is very good. Their neutralizing antibody quantity-wise is also very, very good. So then we go to another example, and how these vaccines are being taken and what risk is being taken. Now there is a Shim5 adeno and the spike protein vaccine, of which is very famously known as AstraZeneca and Oxford vaccine. So AstraZeneca and Oxford has taken a different kind of strategy. So this strategy is this, that give the license to several companies. So they have given this license to Serum Institute of India, and their strategy is this, that if the vaccine if, by the time the vaccine goes to phase 2, and if it succeeds in phase 2, they will start manufacturing. So they will take a risk. They will manufacture billion of doses, and if the phase 3 is successful, they will then release the vaccine as a emergency vaccine. You know that if it is 1.5 year vaccine, it will still be emergency approval vaccine. It will not be a full approval vaccine. For that, you need to do many more things. So this is the catch there, that they will start making it. They are taking this risk. The BARDA and the other government agencies are supporting them. They are taking up the cost. If we do it in India, we need to see that whether there will be any pre-commitment from Indian government to buy this vaccine or not. But there is another big catch here, that when the Oxford, that is why I say that all the data should be out in the open, so that people could make an analysis, and many of them are putting these data in the open system. So the Oxford guys, when they did the monkey challenge, the monkey couldn't get rid of the COVID infection. It became milder. However, they developed neutralizing antibody. So these guys, they were happy with that even if the viral load is suppressed and it becomes a milder infection, but neutralizing antibodies are produced, then we will go ahead. So they are going ahead, and their vaccine is being tested in Europe, in Brazil, in many countries. So this is the second most important vaccine which is going on. This is a tested platform. Oxford vaccine unit is a very old vaccine unit. They have tested platforms which they use. So you have now understood that it is not very easy to really look at that whether a vaccine will succeed or it will not succeed. So the third one, I'm just taking those which are also being developed in India. The third one which is being developed in India is again is going into phase 3 in Australia and Brazil. This is known as Novavax. Novavax is a vaccine company in Rockville. They have made RSV vaccine for the mother to child protection, and this is going into phase 3 in USA. They have also, in their same platform, they have got influenza vaccine. They are using an adjuvant. This is known as matrigel, and this adjuvant is also very good adjuvant. So with this, they uh, they have produced this vaccine in the same platform with the same kind of experience which they had got, and they are funded by BARDA, and they have also done in Australia and are going into Brazil for phase 3. They have also completed now. They're going to complete phase 2 very soon. So there is an Indian company which is known as Cadila. Don't confuse it with Zydus Cadila vaccine, which got approval for moving forward. There's a Cadila. This is known as CP Biologicals. There's a joint company owned by Novavax and the Cadila Pharma, and they are waiting. These guys are not at the moment committing. They are, because it's a lot of money, and if it fails, then you sink the money, and because you have to create a facility, and that facility then goes waste. So this particular thing, they are waiting. They say that if they get success, since they already have vaccines with this platform in the market, they will be able to produce this vaccine and will be able to go to market very easily. So there's another strategy which they have taken, and there is an Indian company with a platform which might move forward. Then the at the moment, the 19 list which are moving forward, there is no mention of any Indian company at this given moment on that list of 19, which are prospective, which is a global list. So we should think of that. Similarly, there is Adeno 26 platform of the Johnson and Johnson. Johnson and Johnson has also moved, and they have the assays, and they have one major advantage over others, that their Ebola vaccine was successful in this platform. Ebola is also an RNA vaccine, not very easy to produce vaccine, but they have tasted success. Their RSV vaccine is also moving forward. They, at the moment, they do not have any agreement with India, but they are at the moment talking to Aurobindo. So Aurobindo has got a new vaccine plant in Hyderabad. So they are talking to Aurobindo that whether when it is ready, whether Aurobindo will take it forward or not. Aurobindo also has a, a, a vaccine company purchased from Pfizer in the US known as Perfectus. So this is also a very strong thing. But Aurobindo also has vesical stomatitis with virus platform, and this platform is also very nice. So the Bharat Biotech's vaccine, which was in the newspaper, is a whole cell attenuated vaccine, a whole cell killed vaccine. So the whole cell killed vaccines may have, there will be 2 or 3 challenges. That if it is alum adjuvanted, because that is the adjuvant which most people use, because you don't have to go through a lot of adjuvant research. It might produce immune responses, but the immune responses normally, if it is an attenuated virus, it causes a mild infection, it can produce a better immunologic response. So at this given moment, it has not gone to human, so we do not know how it will behave. But there is an example, the Sinovac vaccine of China, which is a formalized COVID-2 that is inactivated now because it has been treated with formalin and it is alum adjuvanted, has already gone to human and is moving very fast. This will be the first vaccine in the world which might be available earliest than others. And they also are doing it in multi-country. So in India also, it will not be possible. It will depend on where, when we enter phase 3, it will depend where we are in the phase 3. If we have very little infection by that time, which is not happening at the moment, but suppose if our infection rate comes down, our size of the phase 3 trial will be huge. And most vaccine products are being tested in the multiple countries. So I can go on and describe to you the different vaccine candidates, but I restricted myself to what is really the regulatory pathway, which is not very easy. For this, FDA had to take out a special regulatory pathway. I hope that we will follow and we will study that very in depth, and we will be able to follow that forward. But the uh, the requirement of doing the many of the vaccine, you don't need to go to monkey study. Some you need to go, but like polio, etc., if it is like polio, you have to go to monkey. But for many, you need not go to monkey. But here, it is important that you go to monkey, and this facility at the moment is very difficult to do it in India. It has to be done in phase 3. Bharat Biotech has a BSL-3 animal facility. The ICMR has a BSL-3 animal facility in Pune, but there are very few BSL-3 facilities to do the challenge studies in animals. There is another animal known as ferret, where also you can perhaps do the challenge, but again you need BSL-3 facilities. And in India, the regulatory pathway to monkey studies are very, very long. When you will have to wait about, because this is given by a special ethics committee and the CSPCA, which actually is the regulating body for animal experiment, has to bless the monkey studies. That just cannot be done by the local ethical committees. And the same way the specialized mice is not there. Hamster is another animal where you could produce infection, but it is self-limiting infection for 14 days. But it has some asthmatic symptoms and other symptoms which happen in hamster if you infect with this. But again, for animal studies, you may have to go outside India. That is what Genova has done. It had gone outside India and got it done. And as I say that you will have to do this outside India. Now there is a catch. We do not know at the moment what is the protective immunity. Like I will be very quick now so that you can ask questions. The plasma therapy has opened up a lot of new information. Now, in the plasmas, when you measure neutralizing antibody, which is actually the major indicator whether you will be protected or not, very many convalescent plasmas have very little neutralizing antibody. Some of them have almost no neutralizing antibody. And even when the neutralizing antibody is produced, neutralizing antibody amount is not the same as it is produced in other infections. So this is a catch in plasma therapy. We have now a paper which actually analyzes different plasma therapies, but many of them have been inconclusive that whether they protected or whether they didn't protect. And as you know that when you give plasma therapy, you also give other medicines also. But the point which I'm trying to make that the amount of neutralizing antibody and the quality of the neutralizing antibody which the vaccine produces will be also very, very critical. There might be other parameters, like another parameter which is very important in the COVID production is the NK cells. NK cells are the markers for the innate immunity. And it has been found that if your NK cells are okay, then you are better. And so at the moment, it's a learning process. If you really ask me that that I will produce the first vaccine, I will say I don't know. Even if I produce, I don't know whether it will work or it will not work. So the situation should be like this, that it should not be that there is a vaccine somewhere which has succeeded, which is not available to people. This happened with the remdesivir. Like, like the entire production of remdesivir was blocked by US for its own use. They couldn't take it out. So what the Gilead did that it licensed to 4 other Indian companies, Cipla, had Hitro, the Mylan, etc. So this was available in India. Indian CROs could produce this particular thing. So there are models where the like Novavax model, the Serum Institute model, and some other models where some of the international vaccines will be quickly manufactured in India, and it is possible. So we need to look at these models so that anywhere a vaccine succeeds, we are successful. To make another challenge which comes in the vaccine trials is the phase 1, phase 2 are okay because they are done in small number of people. Many times they are hospitalized and the samples are taken and it is done. But the efficacy phase 3 study, you will have to go to field. You should have a very strong surveillance data in that field area. If you don't have good surveillance data, then you are at a disadvantage. Another thing, so it takes time. What I am trying to say that to develop a vaccine, it takes time. There are no shortcuts in the development of the vaccine. Nobody can, and as also I say that nobody can predict whether vaccine A or vaccine B will succeed or it will not succeed. Finally, there are two more things which we need to understand, that the mutation is happening all the time. In the early period, mutation was not very, very critical. But at the moment, mutations are happening little rapidly. In influenza, it happens. And the WHO every year, because of that surveillance data, is able to advise that these will be strain change. And the regulators around the world, then they accept when this strain change is done. And there is a very shortcut in really making this strain change and taking the vaccine forward. Though here, some of the another thing which people are developing are the monoclonal therapies. But there is no single tocilizumab which is being taken as a monoclonal antibody therapy is actually neutralizing the inflammatory cytokine IL-6. It is not a really therapy which is actually neutralizing and stopping the entry of the COVID-19. So it comes out that one single antibody may not be able to do the function. So people are creating cocktail of antibodies. And at the moment, most favorite preparation is a cocktail of 3 antibodies. So again, we do not know that if the mutation occurs, because we have taken different parts of the spike protein, some of the things, that whether it will succeed or not succeed. So if I am a god, I will be able to say, I wish I am a god, and I'm able to tell you that the vaccine will come on this particular date. Aspirational date at the moment is the way things are going, that sometimes in 2001, either January, February, or March of the 2001. But these guys have actually progressed a lot. Whereas they started working from the 0 day, and they have done the preclinical studies. Many of them has completed phase 1. They have moved to phase 2. Some of them have completed phase 2, and they are moving to phase 3. And they started simultaneously recruiting the phase 1 and phase 2. The problem of a country developing a good vaccine is that they should have good experts who are the toxicology experts, vaccine platform experts, as well as vaccine assay experts. We should have this expertise is not very easily seen around the world. I do not know, but in my 50 years, I will be able to say that our country has enough expert. Vaccine trial people are different. Vaccine manufacturing people are different. Expertise is different. It is totally different. The toxicology guys who can really look at toxicology are different. Regulatory guys are also very, very different. It's not a very easy thing to make a regulatory decision over here. So one of the things which we need to do here is to bring in international expertise if possible when we are developing the vaccine and making decision about vaccine. In my opinion, although I may be contradicted and people will say that I am egoistic and I'm making such statement, in my opinion, we do not have enough people with enough expertise in our country who will be able to, and I should not sound unpatriotic. I'm a very quite an old person and have seen what we have. We need we need to ramp up our expertise in the regulatory and other arenas by getting appropriate internal expertise wherever we are needed, wherever they are needed. And we should have very strong surveillance guys over here and very quickly set up a surveillance system if we really want to get a good result out of the vaccine trials. So I think I am finished. I told you that what will be the challenge and whether there will be a date, but now the government of India has come up with clear-cut statements that they are not expecting a vaccine before 2001. And also our Honorable Health Minister, who has good knowledge of vaccine because he was a polio warrior, has stated that he cannot predict when we will have a successful vaccine. Please add successful, because that is what we may have a vaccine, but it may succeed or it may not succeed, or it may be good or it may not be good. Thank you very much, Dr. Agarwal.
Thank you, Dr. Ganguly. It was a very wonderful and lucid update on what we have discussed in the past. Successful vaccine is a new term. Vaccine, yes, by 15th August, you may be able to produce a vaccine. You may be able to produce a vaccine even today. But successful vaccine will be the one which will prevent not only inflammatory, but also thrombotic and immunological reactions, both short-term and in long-term. If I have to define...
Dr. Agarwal, sorry to intervene. We should not be unkind to ICMR. They really didn't say that 15 August they will have a vaccine.
No.
This was a play of words. Actually, perhaps what they meant that the first shot will go into human if the ethics committee, etc. work. And ethics committees are also a different kind of a bodies. They don't really very quickly, they can sit, because if there are 8-10 centers, to get all the ethics committees to sit and make a decision, it's quite horrendous. So ICMR didn't say in 15th we will have a vaccine.
What I meant was a successful vaccine is the one which will prevent inflammatory thrombotic and immunological reactions, both that means early and delayed. So if you prevent early and delayed immunological reactions, inflammatory reactions and thrombotic infection reactions, then only we will say this virus, this vaccine will be successful. Dr. Ganguly said that in certain segments of the society in US, 25% of the population has been infected. We have the data from the New York, which says around the same thing. And I am personally of the opinion that a large number of, our prevalence of antibodies, seroprevalence of antibodies in Delhi and in Mumbai may be very high, even though the correct data is not available. So therefore, Delhi and Bombay probably will not be the states for a phase 3 trial, because we have already crossed a large number of infections there. So I'll request comments from Dr. Narottam Puri wanted to ask something. Dr. Narottam Puri.
Professor Ganguly, thank you very much, sir, for a very lucid description. I just wanted to know a couple of things, perhaps they're very basic for you, that from the point of view of safety, is one type of vaccine that is being currently developed better than the other? You have mentioned about some new type of vaccines about which their safety records aren't really well known. That's number one. The second was, does the safety alter with age? So what is safe for, let's say, a 15-year-old boy? Is it equally safe for a 65-year-old? And last but not the least, amongst the vaccines that are being developed, what would be your estimate about the need for the frequency at which they need to be taken? For example, for pneumococcal, we take once in 5 years, for influenza, annually. Thank you, sir.
You have asked all very relevant and good question. The safety of the vaccine, because of the very nature of this infection, was a concern right from the 0 days. Like the virus itself doesn't really kill that much, but it produces a cytokine storm, it produces an inflammation, which is immunologically led inflammation. It attacks organs, various organs, again through an immune system. So really, if you really go to really dissect the safety of this is a really big challenge. That is why most of the vaccine manufacturers try to take as little of the virus antigen as possible. So when they got the entire spike protein, some have entire spike protein, some do not have an entire, they have a small part of the RBD, that is the receptor binding domain. Or somebody has only a messenger RNA or 3 or 4 messenger RNA construct as Pfizer has got, which perhaps are most critical. So that is why they are trying to reduce. But when you take a whole virus, when you take an attenuated virus, you do not know. You have to go through the entire gamut of the preclinical toxicology to really know whether that will be working or that will not be working. So, so the toxicology is one of the most complex part of this, and we should not cut any corner in doing the toxicology test. And that is why US FDA has come out with a very lucid guideline that what we need to do in the toxicology test, or what we need to really do before we go to human, to understand that whether this particular thing is worth taking to human. Because when you take to human, essentially you do some routine mice and other studies, two rodent study and other studies, and you think it is safe. It, for the immunologic substances, this this is never like that. Like the CD4, one of the CD4 thing which came for the AIDS and others, killed 6-7 people when it was given, because in mice it didn't produce the cytokine storm. In the humans, it produced it. Now you talked about the age differences. Age differences is very, very critical in this vaccine trial. That is why people are taking classes of 3 different ages. Now, like children have a immature immunologic system. So there, although we have some Kawasaki-like of symptoms in some children, that is vascular inflammation all around, but it is not has yet been directly linked to COVID. Although it produces coagulation defects, it produces inflammation, of the vessels, it produces thrombosis, etc. But in children, this is this is not so frequently seen. But as you grow in the age, this is one very strong reality. I don't know those of you, in our time, we had a book which was known as Love and Bailey's book. The Love and Bailey's book describes these kind of aberrant immune responses, particularly those which cause vasculitis and others, in uh, temperate countries, countries like America and other countries, and they said it is lesser in tropical countries. And that is very right, because that is why perhaps our mortality is little lesser than others. But what I want to say here, that more older people are more liable to develop autoimmune reaction or reaction against its own body. So we need to have a safety thing when we go to older people very, very carefully. Your third question was, you asked all 3 very nice questions. Your third question was that when we need to uh, immunize ourselves, will it be every year, will it be 2 years, or will it be 3 years? At the moment, if you really ask, nobody knows, because it's a baby virus. The entire experience with this virus is of 6 months. The virus produces antibodies, and those which have neutralizing antibodies are so-called protected. So we know this happens, but it doesn't uniformly happen. The titers itself are not very, very high. So although ELISA titers could be high, so what is the guess is this, that like influenza vaccine and other respiratory infection vaccine, this vaccine will have to be taken every year.
Thank you, Dr. Ganguly. I just want to ask, when we talk about the components of vaccines, so what are the companies, so let's say, which are the vaccines being produced using the killed vaccine, then live attenuated, then mRNA, then membrane, then envelope, then spike, then conjugate, and vector. If one example each, you can give it to us.
Yeah,
Which company is working on killed vaccine?
I have given one example each. I have given Sinovac, another vaccine, which is the whole cell inactivated vaccine. Inactivation process is different. We have talked about attenuated vaccine, which is again a Chinese vaccine, and India also. There is a partnership with that. I talked about messenger RNA vaccine. I didn't talk about the in vivo vaccine, which is the DNA vaccine. DNA vaccine is another vaccine, which is being developed in Korea, and is the company is not a Korean company, but they are undertaking trial with the International Vaccine Institute, and this has been funded by CEPI. This is also moving as desired, but this vaccine is not also in the market. I have described the other vaccines, which are the different platforms which are being used. So for the vaccine, the platforms are the main platform. So I told about some other platform, like India's Aedes Candila is trying to use a measles virus platform. They want to make it a vaccine which could be given in the EPI. So as I told you, that Aurobindo with their prospectus have vesicular stomatitis vaccine or alpha virus vaccine platform. So these platforms are known vaccine platforms. Different companies use it. Then different companies have a strength in their adjuvants, like GlaxoSmithKline Beecham they have also come into a partnership. And so they have a series of adjuvant which are known as ASO 1, 2, 3. Their adjuvant is one of the most powerful adjuvant and have gone to humans. And you need lesser antigen and when you use their adjuvant. So the cost becomes lesser. So the cost of the vaccine, which you didn't ask, cost of the vaccine will again be something very challenging. Many people are making statements that they will make it non-profit, they will not enhance the cost. It is not going to happen like that. Cost of the vaccine comes down eventually when more and more people start producing that particular vaccine or platform. But you normally it will not be very cheap vaccine. But I must make it very clear, it will not be a cheap vaccine. It has to be supported by governments. It has to be subsidized by government. It has to be there has to be pre-commitment by government. And let us see that we do not have too many vaccines. We will not know what to do really. If all the 200 vaccine developers become successful, what will you do, Dr. Agarwal? I gave you 200 vaccines. How will you choose? So, and this is not also going to happen. There are going to be few vaccines which will become better, and then you may be able to make a choice.
So I'd like Madam Meenakshi and Dr. A.K. Agarwal to make some comments before we close, before I ask Dr. Kalra to close.
No, Dr. Ganguly, you have given us a real, walk through and talk through this, conundrum surrounding the vaccine. And we are all equally interested, scientists and non-scientists, medical and non-medical persons. And certainly we needed some light to be thrown on the multiple efforts that are going on within India as well as globally. And this letter from ICR, threw the cat among the pigeons, but just as well, because now a lot of things have gained clarity. Thank you so much. And thank you, Dr. Agarwal, for organizing this meet today. Very much required.
Thank you, Dr. Ganguly, for bringing the entire encyclopedia of vaccine to us. Thanks to Dr. K.K. Agarwal and Dr. K.K. Kalra for bringing Dr. Ganguly to us. I'm lucky enough to interact with him on other platforms, but his entire knowledge is really useful. There are two questions from my side.
Me and Dr. Agarwal interact very often, almost every other day.
There are two questions from my side, and you use the word emergency vaccine, and Dr. K.K. Agarwal used the word successful. I would like to combine the emergency and successful. So in case of emergency vaccine, how much percentage of success you would like to bring together? Suppose I say this vaccine is 70% successful. 60%
70% successful, Dr. Agarwal, is a very good vaccine.
Very good vaccine.
This model, Dr. Meenakshi Datta Ghosh will realize, because she was in the AIDS arena. Harvard has created a model that if the vaccine protects 30%, it will not be a good vaccine at all. If it protects 50%, it will prevent 75% transmission. If it protects 75%, it will stop the transmission altogether. So, and because rest of the things will be done through the herd immunity, which will pass from one individual, vaccinated individual, to the other, or the herd immunity of the population which is existing. So this is what at the moment we think about it. Yes. Emergency vaccines are made when there is a pandemic going on, and you have no choice to wait for the entire vaccine development pathway.
Don't you think we are in that particular?
We are in that. So for Zika, this was attempted, but luckily Zika went away. And Zika was also a very difficult vaccine to make. For Ebola, this has been done, and Ebola vaccine was given to the population very quickly. But whatever you do, whether you are a magician or you are not a magician, The emergency vaccine development also takes about 12 to 18 months. But actual vaccine development, when you go through everything in a very minutest detail and all experiences and phase 4 experience and everything, that takes around, previously it used to take 7, 8, 9 years. Now it takes around 5 years if you are lucky, or little less than 5 years, because many of the things are now known, like people use their own platforms with which they're familiar, they use their own adjuvants. Sometimes there is already a surveillance sites available and you don't have to prepare sites. So all of these are possible to contract the time, but no way of, if you do the proper toxicology and everything, you can contract the time to less than 12 months to 18 months.
My second query.
And that will be emergency vaccine.
That's right. My second query, Dr. Ganguly, which I'm slightly confused because perhaps I'm not the expert, but the increasing perception and the need for utility of plasma, convalescent plasma therapy. Perhaps we are too immature to comment upon it, and as well as our all the donors of plasma are within a phase of a minimum possible period, whether it is 28 days or, but there is hardly any neutralizing antibodies, as you also mentioned. But gradually, after a couple of weeks or couple of months, that plasma will become stronger and stronger with more and more neutralizing antibodies present inside. Don't you think it will also help us in?
No, why the 20 magical thing in 28 days is being taken?
Yes.
That is the time we expect the weak antibodies to be there in the person after the person is infected. Second thing, that virus itself can remain up to 28 days in some of the people. In China, some of the things have even seen it at 30 days. And one of the blood transfusion facility in China has reported viral RNA in 5 or 6 individuals from who had donated blood for transfusion. They have detected RNA signature in them. But the problem is that we do not have the neutralizing antibody assay in with us at this given moment. So normally what is done is you do the ELISA and other antibody assays, the quantitative antibody assay, and see whether the titer is good or not. Because titer is also determined. Neutralizing antibody titer should be around 162 or around minimum around 640 or minimum around 160 or 320, or on good if it is 640, not so good if it is 1 in 40 or less. But these neutralizing antibody assays are not available with us. So we do it like that, and then we pray to God that this will work. I will send you, I have analyzed all the data which has emerged from, if you ask Gautam, he will send you. He has got
I
this
will.
analysis.
Thank you, sir.
So if you will get all the data which are in the public domain on the plasma, in the convalescent plasma trials, it doesn't look that good if you really ask me. And so because it's very difficult to really assess also, because they are getting now everything. Like Minakshi ji sent me a name, a VIP name. Now what was he getting? He was getting augmentin. He was getting
That's right.
he was getting doxycycline. He was getting dexamethasone. He
And ivermectin.
He was getting ivermectin, and he was also getting favipiravir. Okay, so he was getting every damn thing. He was a VIP, so nobody took any chance. At the moment, remdesivir was not available in India. Now it is available. He could have got the remdesivir also. So this is how we are being treated. Now suddenly there is a paper by the Ford that they are Mr. Trump's favorite organization, they came out with hydroxychloroquine works. We now know that there's enough data, so WHO also withdrew and other countries withdrew. But if you look at this, this was an observational study, and all these guys have also received dexamethasone. So you do not know what actually worked. So I have not seen that somebody only got the plasma transfusion and didn't get others. But few studies have shown something, but I will send, if you ask Gautam, he will send it to you. I analyzed and created, and also few papers which have come out on the plasma therapy. So Dr. Agarwal, it will be available to you if you ask Gautam.
Thank you.
Thank you, everybody. I'll ask Dr. Kalra to propose a vote of thanks.
Thanks. I first of all thanks Dr. Ganguly for accepting our invitation to come and talk at short notice. Thank you very much, sir. And then I thanks Dr. Arun Kumar Agarwal, Dr. Nakshidutta Ghosh, Dr. Alex, and Dr. Sanchita, Dr. Sunila Garg, Dr. Atul Mohan Kocha, Ira Gupta, Dr. Jack Kishan, and Dr. Jailal for attending this. And also again, I am thankful to Dr. Upasana Arora. I think probably vaccine probably will be available in the first quarter of next year. And still we do not know effectivity, how much it will be effective, whether it will be universal uses or only for vulnerable group first. Let's continue creating awareness among the masses for using standard precautions. Lockdown is not the answer. Everyone must continue to use standard precautions. Thank you to all. I invite everybody for the next Saturday, same time, and with some different topic. Thank you, everyone.
Thank you, Dr. Kalra, Dr. Agarwal, and
thank you.
Thank you all.
Thank you.
Everybody enjoy a cup of tea in your own house.
Oh, very funny. Please commission Zomato in the next meeting.
Yeah, you will get, if you are using Citibank card, for that you will get 20% commission. And
Yesterday, one moment. No, one moment. Please do not discontinue the meeting. I want to share something. Last night, there was a very brilliant interview on NDTV by Pranoy Roy with the Iranian head of Uber. So Uber, the CEO of Uber said, our business in Uber has been very badly affected by the COVID globally. So now 50% business is on Uber rides and 50% business is on Uber Eats. And Uber Eats in India, we have tied up with Zomato. So anytime you feel like eating or drinking anything, just go to Zomato.
Thank you, everybody. Thank you, sir.